Hello, everyone, and welcome to phyloseminar.org. The current theme is state-dependent speciation and extinction models, and this is the third talk in a series of 3 talks on that topic. Please use the YouTube live comment box to ask questions. Today's speaker is Käfer. Jos has always been interested in plant diversity, but has also enjoyed and still enjoys studying the fields of theoretical biology, genomics, bioinformatics, and biophysics. In particular, his recent research interests have been the genomics of plant sex chromosomes, as well as the evolution of reproductive systems and their consequences. Jos is currently a CNRS research associate at the Institut of Evolutionary Science of Montpellier. Welcome, Jos, and thank you for participating.
Well, thank you, Eric, and also thanks to Rosanna for putting this series together. So yes, this is a third talk in a series of 3 on state dependent diversification. So there were 2 fantastic talks already by Sally Otto and by Rosanna Zidel-Ferguson. And I feel very honored to be able to present something after their talks. So Rosanna and Sally presented the methods. So they're very smart people that have contributed lots of methods. And I feel much more like a fool myself after their talks. But I guess that's okay. I'll just have to accept that. I'm trying to ask some questions also about diversification and the models we use. I know it's easier for a fool to ask questions than for wise persons like Sally and Rosanna to answer them. But I'll just take the role of the fool now, and I'm going to talk you through some applications of diversification research I had, and some of the questions that arose from this. So here we go. As Eric said, I'm interested. I'm a botanist. I'm interested in the diversity of plants. So here we just see some flowers, some photos I had on my computer. There's an incredible diversity of flowers, of plants. And we're interested in, like many biologists, in understanding how this diversity came around. So I'm going to focus mainly on flowering plants. Flowering plants are the plants that have flowers. Here is a phylogeny from a few years ago that Suzanne Renner made in her drew in one of her papers. You can see at the right here, the angiosperms are by far, so the flowering plants are by far the largest group of land plants. She noted 260,000 species, but by now it's more like 350,000 species if you look at Plants of the World online or resources like that. So they did have a tremendous amount of evolutionary success, these angiosperms. This could reside in the invention of the perfect flower. The perfect flower is a reproductive structure that unites female and male reproductive organs, so like the pistils and the ovaries and the stamens. However, because we only have one point, it originated somewhere here along the ancestor of the flowering plants. We cannot test this hypothesis statistically. What we can test is how several floral traits influence diversification. So there has been some research on this, and indeed it seems that nectar spurs, like the ones you can see here or here in Aquilegia, bilateral symmetry, like here or in these orchid flowers as well, viola as well, and pollination mutualisms, like in the fig, did increase diversifications of the groups where they arose. And there just seems to be an indication that flowers at least contribute part of the evolutionary success of the angiosperms. So after this short introduction about the angiosperms, about flowering plants, I'm going to talk you through some research. with the question in mind, what can diversification models tell us? So I'll start with one trait, which will be diece as a sexual system. I'll then go to more traits, try to make it a little bit more complex, and then I'll cry out for help, but we'll see once we get there. So sexual systems, there is a large variety of sexual systems in the flowering plants. So the large majority has bisexual perfect flowers with male and female organs in the same flower. But there's also quite a few plants that have unisexual flowers, either on the same individual, like male and female flowers. It's called monoece, and diece, where the female and male flowers are on different individuals. And then there are some stranger combinations with hermaphroditic flowers and female, etc., male and hermaphroditic. So this has been recognized for a long time already, and the first modern classification actually was based on this by Linnaeus. So I'm going to focus on diece. So the separate sexes. It is, as you saw, it's rare. It concerns like 5 or 6% of the species, but it's rather widespread. So if you look at the families of the flowering plants, it occurs in about 50% of the families, and in 51 out of 66 of the orders of the flowering plants. So the orders are depicted here. You can see that the color code is red for the orders that are completely dioecious, and you can see that most are greenish because they contain very few dioecious species. Only these Ambrellales and Garryalis, there are 1 and 10 species, so they're completely dioecious. And the large ones, like the Aspergillus here, where the orchids are, they have very few dioecious species. So diece evolved from hermaphroditic ancestors. That has been even from Darwin made this hypothesis already. There are frequent transitions of hermaphroditism to diece, and Suzanne Renner estimated it would be like 900 to 5,000 times independently in the evolution of flowering plants. And also the transitions are recent. 1/3 of the genera with dioecious species also contain other sexual systems. And probably so diece is derived within the genus or just above the genus level. And this gives rise to what we call sometimes the twigginess of the distribution of diece. So this is a phylogeny of a genus I've been working on a little bit, Silene. It's quite a large genus with a few dioecious species, which are depicted here in red. And you can see, well, they occur in isolated small clades. So their evolution seems to be recent, and their persistence time is short. So the question that has been raised often is why diece is not more common. And well, I'm not going to give a talk about all the biology of diece. I'll just talk you through this quickly. So there seems to be no clear evolutionary advantage of diece in plants. There are better ways to achieve outcrossing, like self-incompatibility. The investment in the flower actually favors bisexuality, because then the cost of producing a flower is shared between the two sexes. And there are several handicaps linked to the cost of males. One of them is that the males don't produce seeds, so you have to find another way to disperse your seeds if half of the population is not contributing. So the hypothesis has been around since the 1950s that diece would be an evolutionary failure in plants. And this was tested for the first time in 2000 by Jana Halbut. So what she did was collect data of dioecious groups. Here the Minispermaceae are a dioecious group, and their non-dioecious sister groups, which is here made up of 2 families. And then she had lots of them, and then she compared those in a sister clade analysis. So in total, she had 94 dioecious clades and their sister clades, and she found in her results that in 70% of all pairs, the dioecious clades were smaller, which was statistically significant, very much significant. So dioecious clades are smaller, but well, these are sister clade comparisons, so they derive from the same ancestor as the sisters. So that should mean that the dioecious clades diversify less. So they indeed have a shorter persistence time in evolution. And this led to the dead-end hypothesis that Jana Heilboet clearly wrote in her paper. And it has inspired many people, not at least because Jana Heilboet, later Jana Famosi, together with Sally Otto and others, set really this new paradigm that Daiichi increases the extinction rate through a series of theoretical and data analysis papers. And it was inspiring. So I even cited a paper I wrote myself, where I was looking for the negative effect of Daiichi on molecular evolution in Silene. But I also decided to have a closer look at sister clade analysis. So let's do this together. So if you have two clades sharing the same common ancestor, can we expect them to have the same number of species? So let's say there's one clade, the red one here, K, and we have N species in total. So the other clade, the sister clade, has N minus K species. And indeed, the answer is, on average, yes. We would expect that both clades have the same number of species, so K over N would be 0.5. Actually, K is uniformly distributed, which has the intriguing consequence that if you take like two clades with in total 100 species, it means that having one species in one clade and 99 in the others is equally likely as having 50 species in one clade and 50 species in the others. So although the average would be that the clades are of the same size, this distribution makes that most clade pairs we look at are asymmetrical, which was already noticed by Ferris a long time ago. But still, if I speak to this with colleagues, they find it somehow counterintuitive, but it's quite easy to do the maths and to see that's actually the case.
And can you just say, I mean, like what is the model that we're looking at here? Like, I mean, that statement has to be associated with a generative model for trees. Is it?
Yes, it's a birth-death model.
Okay.
Sorry, so that's the model that people use mostly. That's widely used in the SSE models as well.
Yep.
There might be different models with slightly different characteristics, but yes. So indeed, this depends on the simplest birth-death model.
Great.
So there is a large role of chance here. And well, of course, the solution is to do statistics. So nothing new here. Well, for instance, sister clade comparisons are a way to do statistics. But then we're going to dive in a little bit deeper again. We're going to simulate them. So that's what I did at some point. I wanted to understand actually what these characteristics of chance would give in the outcomes of the numbers of species in clades. So here I just show a small toy example of 100 species. What I did is use BC not to fit a model, but I used the simulations, the simulations tool that were included in the package. So I simulated trees with two states, one ancestral and one derived state, the Daiichi. So they have both equal diversification rates and the low transition rate. And then, so I did this a lot of times. I did this, what you can do then is select in the phylogeny, you select your sister clades and count the number of species in each clade and each sister clade. So I did this for 300 simulations of 10,000 species, and in total I had almost 30,000 sister clade pairs. And what I found was that the dioecious clade only had 42% of the species in each sister clade pair, and that the dioecious clade was the largest clade in only 33% of the pairs, which is again highly significant. So dioecious clades are often smaller, and on average, there are less species in the dioecious clade, in the derived clade. So what's going on here? We have the derived clades are smaller. We have equal diversification rates. So I was wondering what I did wrong in my simulations. So I went next door to the office just next to mine to speak to Sylvain Mousset, who's a colleague who does mathematics on mainly population genetics, but he was interested in these issues as well. And we figured out what the problem was. So here you have two clades that are selected to be in your study, a dioecious clade and a sister clade, and they have the same ancestor. So if you see that the dioecious clade is smaller, then of course you would expect that diversification is slower here, right? Well, these have been selected, which means that and as dioecious most often derived, that there had been a transition somewhere on this branch leading towards a dioecious clade. So actually, if this transition took place here, then the dioecious clade had less time to diversify and actually smaller just by construction. And indeed, that's what the model that Sylvain made showed. So instead of the uniform distribution of the size of the derived clade, so this goes from 0 to 50%, you have no, sorry, this is species numbers. So this is for 50 species, sorry. So this is 0 to 100%, sorry. Instead of the uniform distribution, you have this distribution in which small derived clades are more likely to occur than large derived clades. And of course, this is not a property of dioecy or of any derived state. It's just a matter that when you look into a phylogeny and you're going to select the clades of interest, you're conditioning on the fact that a transition has happened on this branch to the derived state. If you do this within a phylogeny, then the situation gets even worse. If you just consider that transitions are a random, concentrate process, then the longer the branch is, the more likely the branch is to have had a transition. And so the editor of our SysBio paper here likened it to throwing darts at a phylogeny. Of course, the longer the branch is, the higher the chance it has been hit with the dart or with the transition. So the longer the branch would be, the less time it will have to speciate and to diversify. So if you take this selection bias for phylogenies into account, then you see that the distribution becomes even more skewed. Like there is a very proportionally very large probability of the derived clade to be extremely small rather than quite large. So the species richness distribution depends on the phylogeny and the process that that produced this phylogeny. So we developed a new test that took these biases into account based on permutations of the observed dioecious numbers, species numbers, and the sister clade species numbers. So then we went to dioecy. Because we had shown that it was a pitfall, a flaw in the sister clade analysis, but we had to make a new data set for dioecious species to be sure that actually the clades where dioecy occurred were derived clades. So we went into the literature and looked up all the clades with where dioecy occurred, tried to group them into monophyletic clades with dioecy, and then see if there was good evidence that Daiichi indeed was a derived state. So doing this, we had 115 clade pairs containing almost 9,000 Daiichi species, which is about 50% of the known Daiichi species, and 40 out of 51 orders in which Daiichi occurs. And indeed, we found that Daiichi clades are, on average, smaller than their sisters. So the 35% of the species, on average, in the sister clades were Daiichis, instead of the expected 50-50. So indeed, Jana Halbut was right when she said that Daiichi's clades were smaller. However, if we apply a new sister clade test, this actually is slightly larger than expected under a scenario with equal diversification rates, which led us to state that Daiichi might increase diversification rates. And as you can see here, so here we grouped the number of sister clades pairs we had per order in the angiosperms. So in some orders we had a few, in some other orders we just had only one. And you see there is quite a lot of variation, and just from eyeballing this, so here the red points are where the Daiichi's clade is larger than expected under some threshold, and blue it's smaller, so the result isn't really very clear. You can say on average they're slightly larger than expected, but it's quite difficult to see in which clade actually this is happening. So then at like about the same time, 1 year later, there was a study made by Nick Sabat, but also with a contribution by Emma Goldberg, Sally Otto, Jana Vamosi in New Phytologist. So they used state-of-the-art diversification methods. So they used BC. And if you have seen the previous seminars, yes, you know what it is. You have two states, 1, 0, and each state has a speciation, extinction rate, and there's transition rates between those. So what they did, instead of looking at the whole tree of the angiosperms, they had 40 genera in which the Daiichi occurred and for which they had trees. And then they found, as in their results, so here what they plotted is the posterior probability that the non-Daiichi's diversification rate is larger than the Daiichi's diversification rate. And you see it's a bimodal distribution. So here at this end, it's the clades, the genera where the non-Daiichi's diversification rate is larger, and here at this end is where the Daiichi's diversification rate is larger. And they have two definitions of Daiichi. Either you take the Echina Daiichi and other dimorphic sexual systems in it, or you don't. So they knew, because they have all these wise people in their study, that you should be very careful when interpreting BC results. So they tested this with simulations. So what they took was the observed tree topology, and then they inferred the transition rates between Daiichi and non-Daiichi on them, and set the diversification rates to be equal. And then they simulated this, and then indeed they found the same pattern. You have this bimodal distribution where either Daiichi species are diversifying more or less, and the observed value of what they found is like somewhere in between their simulation results. So actually they found this pattern of having a very high probability of non-Daiichi's groups to have a higher diversification rate. It could largely be explained by tree topology, and so possibly false positives. But this they were able to check using their simulations. So their conclusion was that diece does not consistently accelerate or slow lineage diversification across multiple genera of angiosperms. So quite similar to ours. So that for diece, what you can do with one trait. And now I'll switch to more traits. I'll keep talking about diece. So what is known for diece is that it has been associated with other life history traits. So it occurs more often in trees that have a tropical distribution. It has in plants with fleshy fruits, with wind pollination, with small flowers, and even among parasitic plants. And these correlations are supposed to arise because they help cope with some disadvantages of diece. But if you look at the single traits, these single traits that are listed here, well, not all of them, each single trait has a low predictive power. So I compared the number of non-dioecious versus dioecious genera. So there are about 1,000 dioecious genera, 14,000 in total. And if you look at like a single trait, abiotic pollination, so wind or water pollination, you do indeed see an increase in the number of genera that's dioecious. So 30% of the dioecious genera are abiotically pollinated versus 6% in the non-dioecious ones. You do the same thing with woody genera. It's not completely clear how to define woodiness. So the figures here are not really exact, but you see an increase among the dioecious species, genera. Same for biotic dispersal. But even if you look at like the most significant increase, like 5 times increase in the occurrence in dioecious genera, it's still only 30% of all the dioecious genera are abiotically pollinated. So it doesn't explain a lot. So this led to the question how actually all these traits determine evolutionary success together. Can we have a better predictive power if we combine them and if we study them together? So this was one of the goals we had in our project. We started a few years ago. We were interested in not only sexual systems but also selfing and outcrossing and related traits. So we had a few meetings in Montpellier with our working group. So this is the first meeting. Later, Rosanna also joined, and Sally Otto couldn't make it at the first meeting, nor Hervé Souquet. And this group was led mainly by Sylvain with some help by me. And we had also the opportunity to recruit a postdoc, Andrew Helmstetter, who arrived later. So what Andrew did, one of the first things he did was do a survey of SSE models in flowering plants. So before doing this survey of SSE models of flowering plants, he did a survey of SSE methods themselves, and he drew this nice figure showing the evolution of SSE models over the course of time from 2007, when BC was introduced, with some extensions to quantitative states, cladogenesis, multiple states. And then in 2015, we saw the development. After 2015, we saw the development of these hidden states as well. So Andrew collected lots of data. So he found 152 SSE studies on angiosperms. And in total, because some studies run different models, he had more than 600 model results. So here you see the course of these models, of the number of studies in the course of time. BC was very much used. It led to an enormous increase, but then from 2015 on, it became a little bit less used, and people also started using the HISE models. One of the first things we looked at was the recommendations and the caveats that had been published on SSE models. So one of the first recommendation was by Rich Fitzjohn in 2009, when they included the probability of the possibility to include unresolved phylogenies. They said, well, you can do this, but you should be careful that your sampling fraction is more than 25%. So more than 25% of the species you're actually looking at are in the phylogenetic tree you're running this model on. But there was a methodological study by Davis et al. In 2013 that said, well, if you want to have good inferences, you should be careful that that the minor state occupies at least 10% of all the tips, and also you should have trees with a size larger than 300 to get reliable results. Then, of course, there was this Rabowski and Goldberg paper in 2015 that stressed the propensity of these methods to give type 1 errors. And there's also a very interesting paper in 2015 by Madison and Fitzjohn, who warned against pseudo-replication and how this wasn't really handled in the methods here. So we looked in these data with Andrew what people actually did do. And some of the results were quite appalling. So for sampling fraction, as Rich Fitzjohn said, it should be more than 25%. We still saw that 38% of all studies had a sampling fraction lower than that. For tip bias, it's kind of okay. Only 13% after 2013 didn't follow the recommendation by Davis et al. For the number of tips, it's slightly worse. 43% of the studies had less than 300 tips, and well, this surprised us a lot. Even 12% of all the studies had less than 50 tips. So they're running these models on very small trees. I don't have the figures for the other recommendations that had been put forward, but since 2015, people normally use HISE and do simulations to check for errors. But this recommendation by Madison and Fitzjohn that you should take, you should be careful not to have the problem of pseudo-replication. Actually, we couldn't find the number of replicates, which is the number of independent events of evolution of your trait. We couldn't find this in the studies. People don't report it. So there's no way to check. Then Andrew looked at the data characteristics and wanted to know if trait-dependent diversification was detected depending on characteristics of the data sets. So in gray is where there was no effect detected of the trait, and the colored ones where there was an effect. And you can see here in this one and this one that the larger the trees, then the larger the number of tips, and the older also, the more likely it is that you will find an effect of the trait on diversification. For the number of markers, which is a measure for the quality of the phylogenetic tree, we didn't really find large differences. Tip bias didn't seem to make any difference as well. This one is interesting. So the sampling fraction, so this should be 100%, not 10, right? The lower the sampling fraction, the larger the probability is that your SSE study finds an effect of the trait. So this is kind of a worrying result. What we did is, in the first place, because we were interested in the biology as well of the plants, and indeed we looked at how what the trait types, so reproductive, biogeography, vegetative traits, habitat, pollination, etc., were that people looked on. looked at, and if there was some differences in how often diversification differences were detected depending on trait types. And what you see here is that for most traits, almost half of the studies find no effect, and a little bit more than half find an effect. So there is an inconsistent effect of these traits on diversification if you look at the literature. But of course, we don't know why. It could be a lack of power for these cases where there was no effect detected. It could be type 1 errors in the case where an effect was detected. And also the biological reason we put forward is that there is actually a lot of context dependence, which is also something that we already saw with Diaceae. Sometimes the clade has a higher diversification rate than Diaceae, sometimes a lower diversification rate with Diaceae, and it probably depends on lots of things like what the trait is associated with, etc. So to make this diversification context a little bit more explicit, at the same time in our working group, we also did a survey on the theoretical predictions of trait-dependent diversification. So not collecting the studies that actually used SSE models, but looking at how people thought, modeled, either formally or verbally, how traits would affect diversification. So here is one example. It's selfing, so plants that do self rather than outcross. What you can see from this drawing is that there are lots of ways that a trait will influence either speciation, which is one component of diversification, or persistence, which is the probability of not going extinct. So it's 1 minus extinction. And diversification is speciation minus extinction, so speciation plus persistence. So there are lots of processes that actually can affect the persistence and the speciation, and selfing influences lots of these processes. I will not go into detail here, but you have to see that here in the end you can follow the arrows and then see whether there is a clear prediction that the trait influences like reproductive isolation, which will increase speciation. But for selfing there isn't. For divergence there isn't either. So there is not a clear effect of selfing on speciation. For persistence, neither. So selfing species have they often have larger fertilization success because they don't depend on others. They colonize easier, but evolutionary they adapt less well. So the effect on persistence isn't clear either. So actually, this is the case for most of the traits. The traits we looked at, we found no trait that had like consistent effects on speciation and extinction or persistence in through these processes. So, well, of course, it's quite obvious. Of course, as traits. It's not the traits that diversify. It's the species that diversify. And the species always is a combination of traits. And this combination of traits, to make it even worse, is embedded in ecology and geography. That all influence the probability of a species to speciate, or a lineage to speciate, or to go extinct. And of course, these trait correlations are caused by evolutionary processes. Just to give you some other examples, here is a nice example by Joly and Schoen from 2 years ago. They looked at cleistogamy. So cleistogamy is the fact that some plants produce flowers that never open, and so the fertilization, the pollination, happens in a closed flower. So there's no insects coming in here. The plant does it itself, and it doesn't even bother to open its flower. And they found that this way of this cleistogamy, this way of self-fertilization, happened more often in plants that had zygomorphic flowers, like in this violet. Because zygomorphy is associated to better pollinator efficacy. So the pollinators go to the flower, the, the, the anthers drop pollen on a specific part of the pollinator body, and then the pollinator goes to another flower, preferably from another plant, and then the pistil is designed so it can pick up the pollen exactly on the place where the other flower left it. So it increases pollinator efficiency.
Sorry, I'm a little confused. So can you help me understand? So there's the pollinator comes to the purple part, which is what I would call the flower.
Yes.
Okay, but then the fertilization happens in this cleisto.
Fertilization happens. So fertilization happens in these open flowers, but these open flowers are not capable of self-fertilization. So
Okay.
they produce less pollen, and they're constructed in such a way that they can't self-fertilize. So zygomorphy and these open flowers are an adaptation to increase pollinator efficacy. And if there are pollinators around, then the efficacy is quite high. But if there are no pollinators around, then you have a problem, because you need quite specific pollinators to pollinate those. So, in case there are no pollinators around, the plant also produces some flowers that never open, that self-fertilize as a way of reproductive assurance.
Got it. Perfect. Thanks.
So that was a nice example, and we're also putting together data from many more traits and see how they are correlated across the angiosperms to figure out actually what are the traits going together, how are they associated, and so here we find an important axis, goes from woody plants to herbaceous plants, where selfing is associated to, and like orthogonal to this is a pollination axis with abiotically pollinated plants here, biotically pollinated plants here, and most of the unisexual flowers also, and the dioecious species are here. Well, this is just to illustrate that there are lots of correlations going on. I'll not go into detail. I don't want you to understand this it would require a talk by itself. So traits do interact. So the question then is, how do we test for interaction? So of course, it was Rosanna's talk that gave a very good example. So if you have two traits and you want to test whether it's one trait or the other that's causing the differences in diversification, Rosanna has a good method to do this. But let's go to dioeci again. So here I drew just a toy example with, in red, dioecious clades, and in black, the non-dioecious lineages. Dioeci could be associated to, sometimes it's associated to trees, sometimes to herbs, sometimes to dry fruits or fleshy fruits, which are animal dispersed. It could happen on continents and islands. So here, if you want to test for the interaction of dioeci with all these traits, you would have 2 to the 4, 16 combinations. So if you want to do this properly with state-of-the-art methods, you also have to include a certain number of hidden states. So it quickly becomes quite intractable. You will have a model with lots of parameters. So there are a few other approaches that have been proposed and applied. One is by O'Meara et al. Already in 2016. They had lots of traits. So they here just to show an example with 3 traits, which gives 8 combinations. And what it did is divide this into sets. So here, this would be the set with 0, 0. It might be the dioecious trees, no matter what the third trait is. and then they fit a model for put these together against the others, and then figure out if there is a difference between. They did a busy model, see if there is a difference between these guys with respect to the others. So they did it for all the combinations, and then see in the end where the most significant differences are. Another approach that has been applied and proposed is model averaging. So instead of taking the best model and say, well, this is it, this is the truth, you run several models and average all these models to get per branch rates, and you average these rates per with the weight of the model fit, the model likelihood, or its Bayesian counterpart. So I won't go into detail, just to show that actually there are ways that have been proposed to solve this problem. And if you're interested, I really recommend to read these papers. But then we get to the conclusion that all branches might be different. So each branch might have a different diversification rates. And I find this a little bit problematic, because speciation and extinction rates, they are supposed to be like summary properties, summary statistics, not properties of individual lineages, right? A lineage, either it speciates and then you get two different new lineages, or it goes extinct and then there's no lineage anymore. So for each lineage, you can only have one event. So it's very difficult to calculate a rate just from one event. Of course, branch-specific models have been proposed, and they rely on models that describe how rates change between branches. So not all rate changes are allowed. And then what you can get like branch-specific rate estimates from these models, but then if each branch has different probabilities to speciate or to go extinct, we have to group these branches to calculate the differences. So you can calculate them, you can group them by taxonomy, by phylogeny, or by traits. And this is what in such a study that proposed branch-specific models, what they did by the team of Hélène Morlon. So here, which is colored here, this is in the group of hogs, the diversification rate of these species. You see there's a lot of variation, but then you somehow have to group them together to make sense out of this. So well, I don't know. I'm not into bird biology, so I don't know actually what their conclusion was. But it's just to show that you need somehow to group the species and the lineage together in order to be able to test if there is some differences between traits or groups. So there's also a paper by Sebastian Höhner on these branch-specific models. It's on bioRxiv. I don't think it has been published yet in a journal, in a peer-reviewed journal. So if each branch is different, what would the consequences be? We know that, well, the only stable state for a lineage is to go extinct. It's like individuals. The only stable state for us is to die, right? So then you can define the fitness of a lineage. The fitness is how many daughter lineages it will produce. And this is the diversification rate. We know that traits contribute to fitness. We also know that traits are heritable. So if the traits are heritable and they contribute to fitness, then the fitness would be heritable as well. So this is what you call lineage selection. The fitness, the diversification rate is heritable in a phylogeny. So that means that lineage with a high diversification rate are more fit. So will we get the survival of the fittest lineage only, and disappearance, and the lineage with small diversification rates, will it disappear? Well, I'm into the help part of my talk now. For those who haven't noticed yet, So I'm just exploring the possibilities of what's going on here. So what could we predict if each branch is different and lineage selection is going on? Well, we have to have some scenarios to explain what will happen. So if there's no constraint on diversification rate, that would mean that diversification rates will increase indefinitely. So you start with an initial distribution of diversification rates, and after some time, only the lineage with the highest diversification rates will be left. So the diversification rate will shift toward higher and higher rates. If there is some limits, if there are some constraints on the diversification rates, then you would expect that lineage selection would get rid of all the lineage that are here in the lower part of the distribution. So all the lineage that you will actually see are concentrated in this upper part of the distribution, which is actually and there is less variation than, so the variation is just this, instead of for the whole range that you initially had. So there could be other models as well, maybe density dependence, and probably they need to be explored, and maybe someone could think also still of more models. So what I think the consequences would be, but here I'm speculating, so I'm just calling out for help to see if someone in the audience has good ideas. Of the consequences of lineage selection, I think, will be that most lineages will have similar diversification rates. So instead of having a full spectrum of diversification rates, those with the smallest diversification rates will be lost. So trait or clade associated differences in diversification rates will be minimized. So if you have a trait that isn't as good for diversification as another trait, you will have to evolve other traits that compensate this disadvantage and will allow you to keep up with the diversification rates of other clades. Otherwise, you'll just disappear. So although diversification rates can vary, they will tend to resemble one another more than you would expect just in a neutral model. And the remaining variation in diversification rates will thus be mainly due to stochasticity and external factors. Like if you have a clade that is stuck on some island, there will be less intensive lineage selection, so probably the diversification rates there will be less high than on the mainland. So what you would be picking up with SSE models would be those external factors rather than something which would be intrinsically related to the traits. So my question then is how SSE models can help us answer these questions and explore this. Well, of course, they are very helpful in thinking about these, but I don't know how they would help us in this scenario of lineage selection. So I don't have an answer here. And with this, I'll go to my conclusions. First, about diversification model use. Just some general recommendations. If you use a diversification model, whatever diversification model, whether it's sister clade analysis or the most sophisticated SSE models, you always have to question the underlying assumptions of your model. And if possible, do some simulations using another model that is more complex than the one you're using, or basing it on real data, as I showed in the example of the paper of Sabat et al. on dioecious genera. They used the real trees because they probably are different than the bird death trees that you would expect in the model. Make also sure that you have enough replicates, and we don't exactly know how many replicates you would need to have. So that was something that people ask us every now and then, but we just don't know. I think that the simulation studies still need to be done here. Be aware of any selection biases. So if you're using SSE models to study diversification, well, of course you were tempted to look into your clade of interest, but it actually depends on how you select these clades. Either you take a small clade or a little bit larger clade. This could influence your results. And one of the recommendations we also made in this paper, where we explored where we surveyed the SSE models, is that you should only do this actually with hypothesis on diversification in mind. Because if you're going to explore phylogeny with different methods to look for differences in diversification rates, you will find some. But if you don't have a hypothesis, then you might just be looking at correlations or type 1 errors. There are also a few open questions. So I haven't still seen yet a study, but maybe it has been published, but I'm just not aware of it. I haven't seen a study yet that looks at the type 1 error rates for hidden states models. I also don't know how to account for more heterogeneity than what the hidden states models do, because hidden state models, if you've seen in the previous talks, they give a number of hidden states, but variation in diversification rates could be more continuous than what these models actually allow. And also, and of course, this is a general question for everybody doing statistics, but it's quite often ignored in this diversification research, is how to check for causality rather than correlations. But still, these are open questions, and I'm really excited about possibilities that and solutions that people that would come up with in the coming years. So to end with a maybe more positive note is actually we know by the use of all these SSE models that lineage selection is going on. So these trait-dependent, state-dependent models, they put lineage selection on the agenda again. But we now have to explore its consequences, and I would be very happy to discuss this with people in the audience or later. And with this, I'm just asking the questions a fool can ask, and I'm at the end of my talk. I'll just thank the people who contributed to the work here. So my colleagues in Lyon, mainly Sylvain Mousset and also Gabriel, who was great inspiration for all this work on DAISY. Colleagues in Uppsala and Oslo who did a lot of work in collecting information on DAISY's clades, and then also our highly diverse working group where lots of these discussions took place in the FRB in Montpellier. So thank you for watching, and I'll be happy to take any questions if there are any.
Great, thank you very much. Yeah, that's a fascinating talk. I mean, I couldn't figure out where the source of that asymmetry was coming from, and then between the sister clades, and then you showed that one diagram about the waiting time for the mutation. That was really fascinating and really tricky. You would think that the neutral set that the sister clade analysis was valid, but that was a clear demonstration.
Yes, it's actually a quite simple result, but it's although it's not a SSE an SSE model, I really like to show it because it actually gives you some intuition about what's happening in diversification and what's happening when you're looking at clades. And I also think that's very so playing around with simulations and then trying to do it yourself, it's a very good exercise for thinking about diversification, because it isn't easy.
And so, speaking of which, I mean, it seems like, I mean, you've made a pretty convincing case that we need to be looking at lineage selection models and doing a lot of simulations using them. But what is the state of the art for that? I mean, are there packages that, I mean, you know, do you want to give practical advice for people who are interested in this and say wanting to be simulating under a more complex model than an SSC model? Do they roll their own, or is there something that they could use?
I don't know. So it's actually it's actually my call for help. I'm not as I said, I'm not a specialist on diversification rate models, so I know much less about these than people like Sally and Emma Goldberg and Rosanna and of course Rich Fidgenon, Wayne Medicine have. So I'm just asking to if there are some people have ideas, if there are some studies that I should read and even if you're interested, maybe come discuss. We can try to figure out how to do this.
Cool. One thing that I do find, and you mentioned this a little bit during your talk, I find it interesting that sort of evolutionary success is equated often with rates of diversification, which is interesting because, like, it seems like you could have a pretty successful lineage that's in a static environment where no diversification is really needed. But you do point out that, I mean, there's, you know, extinction is always there. It's always sort of this threatening force. So just surviving and not going extinct is also clearly evolutionary success.
Yes. So, well, indeed, there are clades where there are just few species, but they are very unlikely to go extinct because these few species are so common that although the net diversification rate will be very low, because of the absence of extinction, and they will not go extinct soon. And indeed, you hit a very interesting topic. We're used to think about just in these microevolutionary models, just a lineage as being a lineage, but also some smaller scale determinants of evolutionary success, like how many individuals is this species comprised of, and what is the total area it's occupying. These are also things that one should look at, and what we're trying to do as well. So actually, to couple species areas with diversification rates. So we started to do this a little bit. There doesn't seem to be a good link, but we have to dive into this a little bit further.
Cool. We should wrap up soon, but I mean, I'm just curious, like, what are the potential advantages of diacy, or however you say it?
The potential advantage of diacy.
I mean, I think you said maybe it's less like you have to mix your genome more with other lineages.
Yes, so it is supposed to be a way to increase outcrossing, and outcrossing is a major force in evolution. So not self-fertilization, but outcrossing, and indeed, outcrossing species are shown to have better capacities of adapting to changing environments, for instance, because they have more genetic diversity. So that would be an advantage of diacy. And it has been argued that there are better ways of achieving outcrossing, like having a self-incompatibility system, like the one that Rosanna spoke about last time. All plants have male and female function, but they just can't self-fertilize. So the pollen of one plant should go to another plant in order to have fertilization, and that is a much better way actually to do outcrossing, because in plants that are sessile, dispersing your seeds is very important. So if half of the individuals, as in dioecious, is not dispersing seeds, this is a severe handicap. And of course, in self-incompatible species, you don't have that. You have the advantages of outcrossing, but you don't have the disadvantages of dioecious. So it has been argued
the conclusion is that there does not seem to be an obvious advantage.
I'm sorry?
So the conclusion is that it does not seem like there is an advantage.
Of dioecious?
Yeah.
Well, there's another series of models is based on sex allocation, and these are the work by Charnow and Maynard Smith, that dioecious would be a way to better equilibrate the allocation to reproductive functions for between the sexes. But if you have just insect pollinated flowers, it doesn't make sense. But it could be an explanation why you have more unisexual flowers and more dioecious species among wind pollinated species. There it would make sense actually to separate the sexes, to specialize in one function rather than to have them together. But we don't know how important the contributions of all these mechanisms are.
Cool. Okay. Well, thank you very much, Jos. That was a really fascinating talk. And we're planning the next set of talks, probably going to be about microbial genetics, but they're being scheduled now.
Thank you.
Thank you.